Hello everyone, and welcome to this module related to the Pharmaceutical Technology 2 for this semester. So this lecture is supposed to be given today, the 16th of May 2020. The time of lecture is 12:30 to 1:20 p.m. So let's get started. So before starting with today's lecture, I'd like to talk about pharmaceutical technology as a topic, as a field. As you probably know from the first semester, that pharmaceutical technology is related to the fabrication or development or preparation of different dosage forms. As a pharmacist, we are dealing with drugs, and these drugs are chemical compounds. So the patient, generally speaking, can't take these drugs in their raw nature. So we need to convert these raw materials, these chemical compounds, into a final form that is easily taken by the patient in order to ensure effectiveness and to be convenient for the patient, and at the same time should be safe. So we need to convert every drug into a final dosage form to be delivered to the patient in an effective, convenient, and a safe manner. So that's the general scope of pharmaceutical technology. Now, for the pharmaceutical technology, the development process for any kind of dosage forms, it passes through different stages. So it started from the synthesis of the chemical compound, and then after that approving that there is a pharmacological activity, and ensuring there is no toxicological effects on the patients. After that, after ensuring all these things, we need to convert the drug from the raw kind of drug, from a compound raw material, into a dosage form. That's the purpose of the of pharmaceutical technology. It should be mentioned that pharmaceutical dosage forms can be classified into different ways. So sometimes we can classify the dosage forms according to the site of administration. Like for example, some of dosage forms can be taken orally, some of them topically, parenterally, rectal dosage forms, and so on. So all these things can be classified according to the site of administration of the dosage forms. This is one way of classification. The other ways of classifications include sometimes the type of the dosage form. Like for example, we have tablets, capsules, injections, inhalers, suppositories, and so on. So we do have also a variety of dosage forms, and this is according to the type of dosage forms. Another classification is related to the physical state of the dosage form. Like for example, if we have solid dosage forms, semi-solid dosage forms, and liquid dosage forms, maybe some dosage forms are in the gaseous state. So it depends on whatever the physical state of the final dosage form. So this is another way of classification. Now this slide will show you some of the references that could be useful for the topics related to the pharmaceutical technology module. So we have the British Pharmacopoeia, the United States Pharmacopoeia. These are the major two pharmacopoeias for the development, for the different types of preparations approved by the FDA and the EMA for use by the patients. And also we do have a variety of references. So we have pharmaceutical dosage form and drug delivery systems by Howard Ansel. This is the first one. And also we have the American Pharmacy and Alton's Pharmaceutics, Martin's Physical Pharmacy and Pharmaceutical Sciences. And also we have a very interesting reference, which is the Encyclopedia of Pharmaceutical Technology. So all these books are very useful if you want to read or if you want to get some information related to pharmaceutical technology as a topic. Not only this, we do have also a variety of research journals. So if you want to publish any article, you need to publish it in a pharmaceutical journal. So for example, these journals include the International Journal of Pharmaceutics, Molecular Pharmaceutics, European Journal of Pharmaceutics and Biopharmaceutics, Pharmaceutical Research, and Journal of Controlled Release. These are all journals that show you an updated information or details about the different types of dosage forms fabricated or developed by the researchers. Now for the different topics covered in this semester, I will depend on these two references. The first one is Ansel's Pharmaceutical Dosage Forms and Drug Delivery Systems by Lewis Allen and Howard Ansel. This is the first one. And the second one is Prowell's American Pharmacy by Lewis Dittrant. So this is the 7th edition, while Ansel's is the 10th edition. You can use any edition basically, but these are the two references that I will be use most commonly to cover the topics related to pharmaceutical technology too. Now what we are going to study in this semester. So the topics for this semester will be emulsions. After completing the suspensions in the last semester, so we are going to continue with the course dispersions. So we will start with pharmaceutical emulsions, and then we'll study lotions, liniments, and collodions. This is the second topic. The third one will be suppositories as semi-solid dosage forms, and followed by powdered dosage forms, and then we will go to the semi-solid dosage forms like creams, ointments, and so on. And the final topic for this semester will be incompatibilities in pharmaceutical dosage forms. So we'll try to cover all these topics in this semester. So just to remind you that pharmaceutical emulsions are a type of disperse systems. And if I ask you this question, what is disperse systems? So disperse systems, generally speaking, are systems essentially composed of two components. The first component is the disperse phase, and the second one is the dispersion medium. So we do have disperse phase and the dispersion medium to form the disperse systems. Now sometimes we call the dispersed phase as internal phase. The dispersion medium sometimes called the continuous or external phase. So we sometimes call it dispersed phase or internal phase. That's for the inner part of the system, and then for the external part we call it the continuous or the dispersion medium of the system. Now an important question related to the dispersed systems, this question is do we have a single type of disperse systems, or we have a variety of types for these systems? So the answer for this question is we do have a variety or different types of disperse systems, and these can be classified according to the size of the disperse phase in the dispersion medium into the following dispersions. The first one is molecular dispersions, the second one is colloidal dispersions, and the last one is the coarse dispersions. So the molecular dispersion is the system in which the disperse phase in the form of molecules dispersed in the dispersion medium. So here we have the smallest particle size compared to the other disperse systems. The second one is the colloidal dispersions. So we have a larger particle size compared to the molecular dispersions, but it's still smaller than that of the coarse dispersions. Now, the emulsions, generally speaking, fall within the coarse dispersions. So that's, I'm talking about what the macro emulsions, not the micro or the nano emulsions. So we'll talk about this later on in the next lectures. But if we are talking about macro emulsions or general emulsions or conventional emulsions, these are falling in the coarse dispersion category. Okay, so it's not colloidal dispersions or molecular dispersions, they are coarse dispersions. Now this slide will show you the properties of molecular dispersions. I'm not going to go into the details of these properties. You probably studied these properties in the first semester, but have a look because we probably need to talk about these at some point. So this slide will show you the properties of colloidal dispersions. We probably need to talk about these properties because some of the emulsions, as I mentioned in the previous slide, like microemulsions or nanoemulsions, are falling within the colloidal size range. So the properties are the following. The first one is that colloidal dispersions in general have a specific movement. They call it the Brownian movement. They can't be solved by ordinary microscope. They can be only seen using the ultra microscope or the scanning electron microscope, sometimes using the TEM, transmission electron microscope. Also, they pass through filter paper, but they will be retained by using ultra filters and semi-permeable membranes. That's because of their particle size. They diffuse very slowly, and the disperse phase has a size range from 1 nanometer to 1 micrometers. So from 1 nanometer to 1 micrometer size range. Some references report that the size of the disperse phase may be within the range of 1 nanometer to 0.5 micrometer. So it depends on the reference. They also have low osmotic pressure, and examples of colloidal dispersions are nanoparticles, gels, natural and synthetic polymers in water, and as I mentioned, microemulsions and nanoemulsions are also examples of colloidal dispersions. On the other hand, coarse dispersions are dispersions that are characterized by the following properties. So the size range of the dispersed phase is greater than 1 micron, so it has a larger particle size compared to the molecular and the colloidal dispersions. They can be seen under ordinary microscope, so you don't need to use, like for example, electron microscope or ultra microscope to resolve or to scan the particles within the coarse dispersions. So all what you need is ordinary light microscope. They don't pass through any type of membranes, even ordinary filter paper, they can't pass through it. they have negligible osmotic pressure, and the examples for this type of dispersions are pharmaceutical suspensions and emulsions. So in this case I'm talking about macro emulsions or conventional emulsions, these emulsions having a droplet size of the dispersed phase larger than 1 micron size. So now we are going to start with the first topic in this semester, which is related to pharmaceutical emulsions. The outline of the series of lectures related to pharmaceutical emulsions will cover the following topics. The first thing is an introduction into pharmaceutical emulsions. This is the first thing. The other thing is the purposes or the reasons of emulsifications. So why do we need to prepare this kind of dosage form, the emulsions? Why do we need to prepare emulsions? So we need to know that. The other thing is the theories of emulsification. So we know that there is an emulsification process, but we need to know some background or understanding the science behind the emulsification process, how we get the emulsion, and then the methods of emulsification, followed by the components of emulsions. So that's another thing. And then as an important part of emulsion, we need to know something related to emulsifying agents, because without these agents we probably it's very difficult to prepare pharmaceutical emulsions. And then we will talk later on the HLB system. So this is related to the emulsifying agents. And finally we will cover the stability of emulsions. So after understanding the basics of emulsions, reasons, theories, and then how to prepare the emulsions, different components of emulsions, and then we will study the stability of emulsions. So all these topics will be covered in this lecture and in the next lectures. So the first thing to start with related to pharmaceutical emulsion is a simple definition of pharmaceutical emulsions. So we need to know what are pharmaceutical emulsions. Pharmaceutical emulsions can be defined as dispersions in which the dispersed phase is composed of a small globules of liquid distributed through a dispersion medium, and the internal phase is immiscible with the external phase. So that's the simple definition of pharmaceutical emulsions or emulsions in general. So there are different questions that could be asked related to pharmaceutical emulsions. The first one: do we have a single size of the globules inside the dispersion medium? So that's one of the questions. And then, do we have a specific number of the globules inside the dispersion medium, and so on. So to answer the first question, so the size of the globules depends on the type of the emulsion. Sometimes we have a uniform droplet size within the emulsion. That's the first thing, and we call it a monodisperse emulsion if we have approximately the same globule or droplet size. That's the first thing. If we have a variety of particle size, as can be seen in the first and the second two images, then we call it a polydispersed emulsion. So we have a monodisperse and polydisperse emulsions, depending if we have a single particle size or we have a variety of droplet sizes inside the emulsion. And in the third images, as you can see here in the to the right, sometimes we have droplets inside a droplet within the emulsion, within the internal phase. So we have a small droplets and then larger droplets and then the dispersion medium. So this is what we call a double emulsion. So we have internal phase and then larger droplets and then the external phase. So we will talk about this in the next lectures. But generally speaking, all these types of emulsion share these kind of properties, which it is generally composed of an external phase and internal phase. The internal phase is a small globules distributed somehow uniformly inside the dispersion medium, and there is immiscibility between the internal and the external phases. Now another question may be asked by someone, what kind of disperse systems emulsions are belonging to? So is it belong to molecular dispersion? Does it belong to colloidal dispersion or cause dispersion? So to answer this question, again, it depends on the type of the emulsion. If we are dealing with macro emulsion, then it can be classified under the category of coarse dispersion. But if we are talking about nano emulsions and micro emulsions, is more likely falling within the colloidal size range. So the answer for this question, it depends on the type of emulsion. Now another important question is related to the purposes of emulsion and emulsification. Why do we need to prepare pharmaceutical emulsions? So we already have pharmaceutical solutions, suspensions. Why do we need to prepare another type of dosage forms like emulsions? So to answer this question, there are different reasons that make emulsions one of the important dosage forms in the field of pharmacy. So first of all, sometimes we need to prepare emulsions as a final dosage forms to be used by the patients. Another times we need to use emulsions as intermediate products for the fabrication of another types of dosage forms. But generally speaking, the purposes of emulsions and the emulsification process can be summarized in the following points. First of all, imagine if you have a therapeutic agent in the form of liquid dosage form, and you want to formulate it or prepare it as a liquid dosage form. So the only one way to prepare it as a liquid dosage form, and you have immiscibility between the dispersion medium and the active ingredient, is to formulate it as an emulsion. So in this case, imagine if you have a drug in the form of oil, liquid, and then you want to prepare it as aqueous formulation, so you need to disperse the oil in the aqueous phase, and this will create an emulsion. That's the first thing. The other thing, imagine that the taste of the active ingredient is not good, is disagreeable by the patient, distasteful oil, for example. So in this case, the emulsions can provide a very good option to formulate these disagreeable taste medicines in the form of emulsion. So you can mask the taste of the drug if it is entrapped in the internal phase of the emulsion. So that's another advantage or purpose for the emulsion. The third advantage is you can improve the absorption of the drugs by formulating these drugs as emulsions. The reason for that is because when you formulate the drug as an emulsion, so you will disperse the drug in the form of fine droplets inside the dispersion medium. So in this case, the drug just need to be partitioned from the oil phase, for example, into the aqueous phase, and then it will be ready for absorption. So the small particle size and the partitioning will make it very easily for the drug to be passed from the oil, for example, the internal phase, the oil droplets into the systemic circulation. Okay, so that's another important thing. Another advantage for the emulsions, because we are dealing with internal phase and the external phase, and the internal phase it could be water or oil, and the external phase it can be oil and water as well. So we have a variety of formulation, flexibility. So we can prepare oil in water emulsion or water in oil emulsion, depending on the type of the internal and the external phases. So it could be applied on the skin, different skin. It could be you can control the release of the drug from the vehicles and so on. So you can prepare a variety of products using this technology by using emulsions as a dosage forms, as a dosage form. The other important point, sometimes we have medicinal substances that are irritants to the skin, for example, or mucous membranes. So in order to avoid this irritation, so it's good to formulate these drugs as an emulsions. So in this case, we are overcoming the problem of irritation. So these are generally some of the purposes of emulsions and emulsification. There are another points, but these are the most important ones. Another important point to mention is what are the routes of administration of pharmaceutical emulsions? Are they restricted to certain type of site of administration, or they could be applied to more than one site? So the answer of this question is that we can administer emulsions in a variety of routes of administrations. So for example, we have the oral route, we have the parenteral route, we have sometimes any kind, it could be applicable to more than one route of administration depending on the intended therapeutic activity of the drug, and if it is local or systemic, and the specifications of the drugs entrapped inside the emulsion, and also the specifications of the final dosage form. So the answer for this question is it could be applied to a variety of routes of administration. Now let's move to the components of emulsion. So you probably asked this question, what are the components of emulsion, or what is the composition of an emulsion? So to answer your question, emulsions are generally composed of three important components. These are the disperse phase, or the internal phase, and the other part, which is the dispersion medium, external or continuous phase, as I mentioned previously. And the other important component is the emulsifying agent, or the emulgent, or the emulsifier. So this is the agent which is responsible about the emulsification process. So the agent responsible about the promotion of the emulsification process. So these are the most important components, the general components of emulsions. So this is, let's say, the non-medicated emulsion, or the principal components of the emulsion. But for medicated emulsions, you probably need to add the medicinal substances, or the coloring agents, flavoring agents, and so on. Sometimes we need to add stabilizers, buffering agents, and so on. But now I'm talking about the principal components of the emulsion. So we have the disperse phase, dispersion medium, and the emulsifying agent. Now let's move to the other part related to the theories of emulsification. If we are talking about the process of emulsification, An important question may be asked: how the process of emulsification happens? Like, for example, what is the theory behind the emulsification process? So the answer for this question, we do have three theories that explain the emulsification process. So the first theory is the surface tension theory. The second one is the oriented wedge theory. And the third one is the plastic or the interfacial film theory. So I will explain these theories individually. So let's start with the surface tension theory. In this theory, we do have the internal phase and the external phase, and then we will add the emulsifying agent. So the emulsifying agent will promote the emulsification process by reducing the surface tension between the between the internal phase and the external phases. So in this case, it will promote the emulsification, the dispersion of the small oil droplets, for example, or the liquid droplets of the internal phase in the external phase, by reducing the surface tension between the internal and the external phases. So in this case, we'll get the emulsified droplets inside the dispersion medium. So that's the first theory. Now, the second theory, which is the oriented wedge theory, is based on the theoretical background related to the fact that the emulsifying agents has two different parts. So it's a polar and non-polar portions. Okay, so in this case, when you add the emulsifying agents to the emulsion, so the emulsifying agent will orient themselves between the two phases. So the polar part of the emulsifying agent will be in the polar phase, and the non-polar will be oriented in the non-polar phase. So in this case, the emulsifying agent will act by this orientation to fix the oil droplets and prevent the phase separation of the internal phase from the external phase. So this is another theory that explains the emulsification process. The third one, which is the plastic or the interfacial film theory, this theory states that the emulsification process is happening because the emulsifying agent will coat the internal phase by a film. So it will coat the emulsifying agent by a film. That's why it will keep these oil droplets separated, and in this way it will prevent the phase separation of the internal phase from the external phase. So these are generally the three different theories that explain the process of emulsification. Now, before ending this lecture, I'd like to mention some information about what do we mean by mono versus heterodisperse or polydisperse system. So as you can see in these images, the first one to the left, we have a very uniform droplet inside the external phase. So in this case, we call it monodisperse system or emulsion, while in the left image, we do have a variety of particle size, and that's what we call it a hetero or polydisperse system. So that's the difference. So monodisperse and heterodisperse is depending on the uniformity of the droplets inside the emulsion. Now, if we try to analyze mono and heterodisperse systems using, for example, the zeta sizer or something similar to it, so we will get like different peaks. So if we are talking about monodisperse system, the peak will be very sharp, small and very sharp. While if we are talking about monodisperse system, then we will get a very broad peak. So indicates that we are dealing with a wide range of droplet size. While if we have a monodisperse, we are dealing with a narrow one narrow range of droplet size. So that's how we will get under the analysis using the zetasizer or any other instruments to analyze the particle size of the emulsion. Now, to summarize this lecture covered the following topics. So it already covered an introduction into pharmaceutical technology too, some of the references that you probably need during studying this module, and then the topics for this semester we are going to cover. And then we moved to review of Disperse systems, and then we started with the first topic related to this semester, which is pharmaceutical emulsions. I already explained what do we mean by emulsions and what are the formulation strategies, components, the principal components of emulsions, and then talked about the emulsification theories. So that's all for today, and I hope that you enjoyed listening to this lecture, and we'll continue with the other topics in the next lectures. So if you have any questions related to this lecture, please send these questions on my email. My email is already on the slide, muqdad.musa@universityofbasra.edu.iq. So this is my email. And I hope you will enjoy the topics related to pharmaceutical technology, and I'll see you in the next lectures. So have fun and stay safe. Bye.